Hey, thanks for the opportunity to talk. I've got some brand new data, and this is the first chance I've really had to talk about it, so thank you. So we're looking at predicting the translation efficiency of 5' untranslated regions. This is a collaboration with Georg Selig's group in the computer science department. Really, our lofty goal here is, can we predict how any cis-regulatory sequence will influence translation or protein accumulation? So knowing any combination of sequences, can we predict the outcome of how that protein is translated? You can imagine this is a giant sequence space, and this is just kind of a mock-up of the yeast regulatory region, which is really nice and compact. Shout out to yeast for being a great model organism. So we're going to focus on, to start with at least, ideally we're going to move to all of these regions, but work with the 5' UTR. The 5' UTR is important, obviously, for pre-initiation complex assembly. There's a few known regulatory components of it, like upstream open reading frames, upstream start codons. But there's really not many known motifs. Very rarely are there binding proteins. None that I know of in yeast specifically, but many in mammalian setups. So we want to be able to use the genetic tools that yeast provides us and this capacity to work with really large libraries and see if we can predict how any sequence in that 5' UTR will affect translation of the gene. So just a quick outline, I'll talk about the experimental design. We'll kind of confirm a sanity check on our assay to make sure the biology is there and kind of extensions of that. And then we're going to talk about modeling of our library using the convolutional neural network. And then after that, the brand new part that we're just still looking at is, can we evolve UTRs to be better translators? So UTRs generally are about 50 nucleotides in length in yeast. Some are much longer, but if you just kind of go with 50 as a round number, and you imagine every possible sequence that combination is a massive number. And so people have obviously looked at UTRs in yeast in other capacities. Our own lab, Matt Rich, did a mutagenesis. This is the SOL1 promoter and selection on sulfur limiting conditions. And kind of the way I think of this is if you have a giant sequence space over there on the right, mutagenesis, you have a really strong point that you're looking at and kind of fades out as you go away from that point. And that's kind of the information you can glean from that little area. Aaron Segal's group has taken very small libraries and sorted them based on YFP, and this is about 2,000 variants only, and came up with some very simple rules. And again, this is a much more saturated, very small region of that giant area. So you could try to look at all of the known UTRs in yeast, but again, that's only 5,000 total. And a lot of these will have similar architecture, and so you would kind of end up with clustering of very similar sequences. So really what we want to do is take a really massive library and try to cover this area in a much more broad, even-spaced way. So how we're going to do this is using growth selections afforded to us by yeast. And really what you can do is you can take yeast knockouts for nutrient markers, in this case His3. You can complement that with a His3 gene driven by promoter. This is just a housekeeping gene, Psych1, relatively low expression gene. We've had success with this in the past. Leah, who's out there right now, used this to assay BRCA1. And Griffin previously had looked at high throughput protein stability measurements. So what we're doing, this is just kind of proof in the pudding that when we have the wild type Psych1 gene compared to Psych1 promoter, compared to when we add a strong GC hairpin in the 5' UTR, we get a big difference in growth. And this is relative doubling time, so a smaller number means faster growth. So we have a big difference by just adding a little bit of 3-aminotrizol, which is the competitive inhibitor for His3. So this is just kind of targeting the best selection parameters. So from there, we're introducing random 50 nucleotide sections right upstream of His3, replacing the endogenous Psych1 UTR with this completely random, about 500,000 random 50 nucleotides. And in batch culture, we're growing these under the selection conditions. and we're doing high throughput sequencing of this large library, as well as 5' end capture to see if there's differences in the transcriptional start site, which I won't have time to talk about today. So we sequence at the beginning and the end, and the difference in the proportion of sequences for any given part of our library tells us the fitness, how well that grew in competition, which is a readout of the translation of history. So just kind of the reason we need such a large library is if we look at modeling these UTRs where we model only based on 1,000 UTRs, this is our predicted growth rate from modeling, versus the actual growth rate. 1,000 UTRs, we have really poor ability to do so, and increasingly it gets better as we add more and more UTRs. So really the size of the library is important here, and that's one thing that yeast really allows us to do. So we can pull these out individually and test them in individual growth assays, spotting assays, and doubling time assays like I showed before. And the correlation with our sequencing compared to the individually tested is pretty good here. So from there, we'll just kind of go over a few of the simple parts of the biology here before we get into the modeling. One interesting thing is if we look at how each nucleotide does per position on the UTR, there's kind of an interesting trend here, which is the dark colors here are the fraction of nucleotides at this position in the native genes. And in the lighter colors is the growth scores for each of those nucleotides on average across all those positions. And you see they mirror really, really well. So that probably tells us that most of the native UTRs are really optimized for the best translation already, which is kind of interesting, and we weren't quite expecting that. The other thing we looked at is the Kozak usage. The most common Kozak here, which is the 5 nucleotides right upstream of the start codon. The most common one here, this is the occurrence in the yeast genome. The most common one actually doesn't have the best translation rate. Several of these do better based on our data, and at least to some degree, that's true using ribosomal profiling data as a look at translation, but the data, there's just not quite enough data compared to what we have. As expected, upstream start codons also have a pretty significant effect, especially if they're out of frame. Anything out of frame per position does really poorly. And with a stop codon, everything does poorly. Without a stop codon, the in-frame stuff still does fine. Having a small upstream region in frame is fine. As you go farther and farther away, it has a larger effect on the total growth here. And so if we just look at these upstream ATGs and kind of look at what the codon usage here is, a kind of interesting thing pops up, and that's this correlation between the scores of the codons in those upstream upstream regions compared to the codon usage fraction in yeast. And this really is a readout of higher codon usage usually means higher translation rate for those specific codons. Really low abundance codons generally are not translated as efficiently. Another component of the biology here is if we look at secondary structure, it does have a small effect on translation, and that's measured by minimum free energy here. However, you really need to get a really strong secondary structures before you have much of an effect on translation. And I guess my hypothesis on this is you have all these initiation factors, and a lot of them are actually helicases. So presumably anything that's not a really strong secondary structure is just being stripped off by these initiation factors. And also if you have these really nice, strong 5 nucleotide through 7 nucleotide hairpins with a loop, you have a pretty significant effect on translation or growth. Okay, so the modeling part of this. So for this, we're using convolutional neural networks. If you're not familiar with these are probably most well known for analyzing images. And kind of the way you can think of this is if you fed this type of model 1,000 pictures of a cat, it would pick out specific components of those pictures that would tell it that it's a cat. So those aren't necessarily just a group of pixels. It's really complex interactions between a bunch of groups. And so similar to just measuring pixels, we can give it kind of a pixel version of our DNA sequences and a score associated with those. And so at the bottom level, this acts much like a positional weight matrix. But as you get farther into more layers, more complex layers, there's more complex interactions and dinucleotide position weight matrices that are much more hard to interpret, but gives us a much better model that's beyond just kind of a simple linear model. So this was the modeling stuff was mostly done by Alex in Georg's lab. And previously, he had done a lot of work with these 3-mer linear models. And so we wanted to compare how the convolutional neural network does compared to the 3-mer models he worked with previously. So this is a 10% held out test set where we're making the model a 90% and then measuring the other 10% of our data. And so if you compare the 3-mer additive linear model here to the convolutional neural network, our convolutional neural network actually does quite a bit better than the linear model. We were pretty happy with this score, with these scores. Like I said before, you kind of get a position weight matrix from this. And so on the first layer, we have 64 filters that you can kind of look at much like those weight matrices. And you pull out several key components here, stuff like start codons are important, stop codons. We kind of expected that. But we get a lot of other filters as well, like a quadruplex Gs. One thing that I just dug up looking through the literature, and one nice thing about working in yeast is there's 30 years of genetics to go with this, is the CAAG motif. A couple early 90s and 80s papers showed that the CAG is important for actually setting the transcription start site for a lot of these. So presumably, we're actually changing the transcription start site with this CAG. There's a lot more biology to dig through with these motifs that we have not had time to do yet. So outside of our giant test set, we also want to know, can we predict how the native UTRs will score? So using our same system, the Psych1 His3 system, we synthesized fragments of the native UTRs, 11,000 total fragments. If a UTR was longer than 50, we would just fragment that up, introduce those right upstream of His3. And this does actually better than on our big test set. These do really well. And part of that probably is because a lot of these translate pretty well. So a lot of the very, very negative stuff might be skewing our model scores a little bit. So from there, what we really want to do is say, can we take a test set, take a random sampling from either the native or the synthetic set that we built, and can we evolve those UTRs to have higher translation? And so what we did is we had a start point here, start score, and we asked the model, what's the best single nucleotide change to increase translation? And we did that iteratively until there was no appreciable gains beyond that. So we did that both for our random synthetic 100 UTRs, and then also 100 from the native set. And one interesting thing you can kind of look at here is you can look at the types of motifs, or in this case, I'm just looking at formers that are at the end and the ones that are at the beginning. And in the blue here, for both the native and synthetic set, you can see a pretty even distribution of the types of formers. And then the green is enrichment for specific types of formers. And just some examples of the stuff that's at the bottom and the top, you see the quadruplex G is very negative, and generally a lot of G-rich stuff here. Generally a lot of A-rich stuff. You can see the CAG is there as well, and some other components. And this is an area that we can really dig into more and have not had the time yet. So to test these evolutionary predictions, we took the start point, the mid-step point, and the end point, and synthesized these all in an array and introduced them back into our same system. Now you notice at the midpoint of the step, the vast majority of the changes have already been made, and many times there's not a huge amount of change between the mid-step and the end step. And so this is really reflected on the results that we got. Here's the starting points. And again, we have a range of the start from very poor to relatively decent in the blue here. This is the actual growth versus the predicted growth. And so you can see that we're definitely evolving them from the start point to the midpoint pretty well and consistently can see that in the box plot here as well. But beyond the midpoint to the end point, we don't see a ton of additional growth. And so that possibly is just because we might be overfitting our model a little bit. But we're pretty happy with this increase. So this is with the random set. And it did work with the native set, but probably not as well. And you can see one major difference is, like I said before, the native UTRs on average are pretty well optimized for high evolving. So ones that started pretty poorly, we could evolve quite nicely, but the ones that started with a very high growth rate, we could not increase that growth rate a whole amount. So the plan from here really is we want to go back to these step-by-steps and see if we're going to synthesize every step and really determine which components of these step-by-step predictions are the best. And then, like I said before, the grand plan here is to take libraries just like these 5' UTR libraries and introduce libraries of the upstream promoter, the core region, 3' UTR, and the introns, and an intron for history, and see if we can combine these types of libraries for one model that would tell us any combination of these sequences, what will be the translation. The other nice thing we can do with this is we can do these types of assays in knockouts for specific types of translation and stuff like that, and really get an idea of what effect on the cis-regulation having a knockout for any component you're interested in. And beyond that, what we will also be doing is doing this in other gene contexts. Like I showed before with Matt Rich's work with SOL1, that's the SOL1 promoter and the SOL1 gene. And so we actually have a 5' UTR library in the SOL1 promoter, and the selection there is in sulfur limiting conditions. So we'll be combining these two genic libraries into a better model, hopefully. And with that, I'd like to thank Stan, our collaborators, Georg, Alex, Ben, and Anna, and Matt Rich helped with the SOL1 data. Thank you.
Any questions for Josh?
I'm wondering if there might be a benefit to trying to pull out, I guess, monosomes, like use an initiation blocker, pull out, or I guess it would be an elongation blocker, try to pull out actually primed ribosomes which have initiated and try to see if this, some kind of sequencing-based approach that would tell you what was really good for initiation as opposed to other steps.
Right. Yeah. I know Georg's lab is working on kind of a ribosome profiling approach to the library, where you would kind of have the same type of library and use the actual ribosomes to kind of measure the translation rate. So I think you're right. There would be some interesting information there. Bill?
For the evolution thing at the end, that's really interesting. I guess I'm wondering whether you're starting from sequences and then sort of making small changes and seeing where you can find, but that's a very local operation.
Yeah.
Have you considered other sort of more bigger steps in your optimization?
Yeah, I think that's a good idea, and we'll probably get more out of that, especially kind of after we plateau. Presumably, we're making the really obvious choices first, but I think you're right that if we added more complex, we still end up with a lot of interesting motifs like that. The CAG I talked about, and several other of those really evolve, and it's not like there was just one change to make those. Many times it's many changes. So, but I think you're right. Bigger blocks of changes might be a better way to evolve beyond, the best. We clearly kind of like maxed out, except for there's a few cases that we haven't really looked at in detail, but 4 or 5 of them we can get much, much higher translation than almost all of the rest of them. So we haven't really quite figured out why those are special yet. Yeah?
Will you try different conditions? Check if it is condition-specific translation efficiency, or if you try it under
Yeah, we haven't done any stresses. You know, there's a bunch of perturbations. You could do it in heat and stuff like that, which I think would probably be the next evolution of this, to see if there's really interesting stuff there. We have confirmed these using YFP, and we're working on this other promoter, the SOL1 promoter too, to make sure this isn't all based on the one context of the single gene. Great, thanks.
Our final speaker is going to be Graham Johnson, who's from the Allen Institute.